last video, I described the organization of muscle, the fascicles within it, and then the muscle cells within those fascicles. So now I'm going to go a step deeper and describe the myofibrils within the cell and spend the most time defining and looking at the sarcomere and the organization of the myofilaments within it. And the myofilaments are going to refer to what we're going to call thick filaments and thin filaments. And the sarcomere is the smallest contractile unit of muscle. So understanding its organization and understanding the physiology within it, which is going to be the sliding filament theory, understanding those two components allows you to see how it is that muscle contracts, how it is that skeletal muscle works.
And in order to get to that point, we need to take a step back and define what a sarcomere is, and also look at the different components of the sarcomere. And in doing so, it'll get us down at the level of the myofilament. So I wanted to start really where I left off in the last video here, and that is in looking at a muscle cell. So this whole thing is the muscle cell. And if you look within that, you see that we have these myofibrils. So myofibril, 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 right? And you have these myofibrils jam-packed within every muscle cell. Another feature that I want to draw your attention to, so you have the sarcolemma here, if you can see. So the sarcolemma, this is essentially going to be the unique cell membrane for a muscle cell membrane. And then another feature that I want to look at is your sarcoplasmic reticulum. So it sounds a lot like endoplasmic reticulum, right? Another organelle that we have learned in the past. And it is a very specific type of ER. So the sarcoplasmic reticulum is shown in blue in this graphic. And if you look around each of these myofibrils, particularly this one and this one, you can see that the sarcoplasmic reticulum wraps around each myofibril kind of like a sleeve. And you're going to have these channels that run the opposite direction. So I'm trying to draw another sarcoplasmic reticulum on this myofibril so you can see a little bit better on the graphic since it's difficult for me to point. But that's your sarcoplasmic reticulum. So this is going to become really significant because this is a place where we can store calcium. And that is going to be the triggering ion in the sliding filament theory. And this is going to be the source of it coming from the sarcoplasmic reticulum. So it's worth pointing out here because this is going to surround each and every myofibril. And it's within these myofibrils that we have what are called sarcomeres. So that's where I want to turn next. Let's take a look at what this sarcomere is. So looking at this image, you can see that there are what are called Z lines coming down here, and there'd be another one approximately right through here. What this division is showing is defined as a sarcomere. The length of one of these myofibrils, right, so pulling one of these guys out, the myofibrils are stacked back to back to back to back. So they're completely adjacent to one another, running the length of the entire myofibril. And so to take a closer look at what the sarcomere is, so I've begun a graphic here, and you can see, so we have one Z disc here running to another Z disc. So these are defined as the borders of the sarcomere. Now in the middle of the sarcomere, fittingly, you have what we're going to call the M line. So M for middle, if you want to think about it that way. And coming off of this M line, you have thick filaments. And each of these thick filaments is comprised of myosin. So let me write some of this down. And the protein that makes each of these up is called myosin. I oftentimes think about myosin almost as a Twizzler. So if you can picture these long strings wrapping around, and then at the end of it you have these heads, my lovely artwork here, but you have these heads called myosin heads that we are going to see play a pivotal part, pun intended, in grabbing a hold of the rest of the sarcomere. And so the other piece here that I need to explain, so we have thick filaments, so I think it follows then that we would also have thin filaments. So all of these blue lines here, and the ones over here too, so each of these are thin filaments. So with the thick filaments, you really just saw this main protein, myosin. With the thin filaments, we're going to see that there are 3 different proteins involved. And I'll introduce those key players in just a moment here. But to recap, so a sarcomere is going to run from one Z disc to another Z disc. And so those are defined as the borders of the sarcomere. In the middle of that, you have the M line. And the M line is going to serve as an anchor point for each of these myosin fibers. And each of these myosin fibers has a myosin head that we will see is ultimately going to attach to the thin filaments. This is also a good place to introduce 2 different bands or patterns within the sarcomere. It becomes a little bit of an alphabet soup with I's and Z's and H's and M's. The main ones that I really want you to know are going to be the H zone and then what we call the I band. And so I think about it as a skinny letter, if you will. And so I band, it is going to run where there are only thin filaments. So if you look over here on this side, so the I band is going to run from here, so following up, you see where there's no overlap with the thin filaments, with the thick filaments, it's just the thin filaments along here. And then same thing along this side. So I'm going to stop drawing this line where we reach the myosin. And so this is just an imaginary line that I'm drawing with the purple. But from there to there, this is going to be what we define as the I band. And so again, this is a region where you don't have any overlap, and it's going to be thin filaments only. So with the H zone, it is going to be a region where there is only thick filaments and where you don't have overlap with the thin filaments. So with the way that I've drawn this particular sarcomere, right, I can draw this, again, imaginary line coming down here where I'm only grabbing the myosin, but it's not a region of overlap with the thin filaments. So from here to here, this is going to be what we call our H zone. So, and then the H zone is a region where it's only thick filaments. So I, kind of a nice tall skinny letter, is going to be thin filaments only, a region of no overlap with the thick. And then the H zone is going to be just the opposite, a region where that's only the myosin and no overlap with the thin. Those 2 zones are going to become relevant when we start looking at the contraction itself. So hang on to that terminology for now. So let's move on and look in more detail at what makes up these thin filaments specifically. So I am now, if you can imagine me, taking a lens and zooming in on this thin filament. That's what you're going to see down here. So let me introduce these key players. So the yellow beads that I've drawn in this diagram here, so each of those are going to be what we call actin. The blue rope, if you will, that twist around, this is going to be what we call tropomyosin. And then lastly, these gray protein molecules, these are going to be what we term troponin. So those are the 3 key players that make up these thin filaments. And so the way they come together is on every single actin protein, there is a binding site. This is a myosin binding site. And I do, in my mind, kind of picture beads on a string, with the beads being the actin, the tropomyosin being the string, And we're going to see that troponin is going to be essentially the key that is going to free up the myosin binding sites on actin. So bear in mind that with muscle, it's perhaps even more important to be able to control and regulate it at rest than it is to be able to stimulate it. So what tropomyosin does is the way that it wraps around these actin, it actually blocks the myosin binding sites on actin, so that our myosin heads here cannot reach up and grab it. And the myosin heads, they have a strong affinity. So these red proteins up here, they have a very strong affinity to reach up and grab the actin on these myosin binding sites. But tropomyosin, again, this blue rope that wraps around, it blocks those binding sites. I've heard an analogy drawn of actin and myosin to being star-crossed lovers, and tropomyosin is preventing them from touching, from communicating, from kissing, what have you. And so the goal is for tropomyosin to move, to shift, and free up the myosin binding sites on actin, so that myosin, I'll clean this up a little bit just to draw it a little bit better. So these myosin heads can actually reach up and grab the actin. And at that point, they will be very content because they can then initiate contraction. So to say a bit more on this, I'm going to zoom in and show a cleaner picture. And the trigger, if you recall, so ask yourself, what is it that was stored in the sarcoplasmic reticulum? And hopefully you recall that it's calcium. So calcium I'll draw as a purple triangle here. There we go. So what calcium is going to do is it's going to come in and it binds to troponin. So it's going to be released from the sarcoplasmic reticulum. We're going to see that an action potential is essentially going to trigger that release, and an action potential that travels down those triads. But for the time being, know that calcium comes in, it binds to troponin, right? It binds to one of these guys. It causes a shape shift in tropomyosin, in this rope that wraps around, and in doing so, it reveals the binding site. So it turns these beads on the string, and it enables the myosin head down here to reach up and grab it. And as soon as the myosin attaches to one of these actin molecules, you call this junction between the two, you call it a cross bridge. So to imagine it up here, again, the myosin would reach out and grab the actin, reach out and grab the actin. Right, so I'm just trying to show it at a smaller scale below and panning out a little bit above. And so again, this would also be called cross bridges. Now, at that point, what we will see happen is these myosin heads, as soon as they're able to grab actin, they are already in a highly energized, cocked, excited position. And so as soon as they reach out and grab actin, you're going to see that they pivot. They have a little bit of a hinge. Let me see if I can draw a bit of a hinge. So if you can picture this hinge connecting the head, I'm drawing right over here, it pivots, and in doing so, it moves towards the midline. And so coming up here, trying to draw, so these myosin heads, you can picture all of these guys grabbing a hold of actin and pulling, and they're going to move this way. And so they're going to pull everything towards the M line. So let me show you a different way of illustrating this. This is the M line here. These are your Z disc. I'll draw them zigzag like I have before. So these are your Z disc or Z lines. You can see either way illustrated. And you can see that as these myosin heads have reached out to grab the actin, they're going to pull. And so this is showing one over here. And so it's pulling. You can see the sequence of events where it moves from here to here. And you can see the Z lines getting closer together. You can also see that as the myosin heads pull, let's think about what happens to those zones that we defined previously. So if you recall, right, what is the name of the zone that, that only has thick filaments and no thin filaments? And so we are seeing that from here to here, right? And this was going to be called the H zone, or the H band. So there's the H zone in that graphic. Here it is here. So there's our H. And then now look how small the H has gotten as the sarcomere shortens, right? So the sarcomere, again, from Z line to Z line, it will get shorter as the sarcomere contracts. Now let's also look at the I band. And so the I band is that region of overlap where you only have thin filaments. And so there would be, another sarcomere coming off of here, and another one coming off of here. But if you just look at what's visible in this, with this sarcomere, so here's the I band. Here it is again, getting smaller, and here it is again. So the I band will also get smaller during contraction. And so I always think about it as saying hi, right? These are the two zones that get smaller during contraction. Another thing that I want to emphasize while I'm on this graphic is that this is called the sliding filament theory. And the filaments, right, are going to be our thin filaments and our thick filaments. And they slide over each other. So if you look at the length of one thick filament, right, from here to here, and here to here, and here to here, do you see that that has not changed one bit? Now we've pulled on the actin, and we've made that shorter. But the length of the thick filament has not changed at all. Now let's take a look at the thin filaments and their length. So to use purple here, so it's going from here to here. In this picture, from here to here, that's the same length from here to here. And you will see that from going from here to here and here to here, and then the same thing on the other side, that the length of those filaments do not change. It's simply that they slide over each other. You have an area where they have increasing overlap, right? That's why our H zone and our I band get smaller, because those were defined as regions where there is no overlap. So they're decreasing as we go. So I want to come back to one final point here before I conclude this video, and that is looking at the role of ATP. So how does ATP come into all of this? Students oftentimes want to tell me that it's involved in what we call the power stroke. So if you come over here and look at these graphics, so this is when a cross bridge is first created. So you're seeing a myosin head reach out and it's grabbing one of these actin beads, right? And as soon as that happens, again, we call it a cross bridge. And I noted before that the myosin head is cocked and in a loaded position. So as soon as it binds, it is going to pivot and pull the actin towards the midline. So the student's gut reaction is to say that this movement, which is called a power stroke, where myosin pivots and pulls on actin, they want to say that the ATP is utilized in this, but that is not correct. And so I love this mousetrap analogy, because if you think about a mousetrap, right, the ATP is invested when you set the trap, when you pull back that spring. And then at that point, the mousetrap or the myosin is fully loaded. It's wound up with a great deal of potential energy, and it's ready to engage in the power stroke. It's ready to snap shut and catch the mouse. So if ATP is not used directly in the power stroke, where does it come in? And you really see it coming in these last two figures here. So ATP is going to be used to break the cross bridge. So ATP breaks the cross bridge. And I always think about it as recocking the myosin head. So it puts the myosin head into this, so cocking of the myosin head, it puts it into a fully loaded position where it is tied to a great deal of potential energy. So again, the first thing ATP does is it breaks this bond. You've got to get the ATP, or excuse me, you have to have the myosin head separate from the actin. And then that ATP is going to be used to recock or to set that spring on the mousetrap so that we can then utilize it. An excellent example of this that frequently comes up in this discussion is that of rigor mortis. So many of you, I assume, have some familiarity with rigor mortis, right? So after death, there's that stiffening of the muscles. And if you think about what's going on, there's no ATP available. So what happens is that we don't have the ability to break this cross bridge. So with rigor mortis, there's no ATP, and we can't move from the picture that's shown here to the picture that's shown here. So the myosin heads essentially remain stuck on the binding sites on actin, and the bridge between actin and myosin remains, and so that locks up the muscle. So these graphics are showing, single heads of myosin binding to actin, but you want to picture hundreds and thousands of myosin heads along the length of the muscle, all binding to actin and all being unable to break. And that's what causes that initial stiffening of the muscle after death.